Alright, hello everyone. Welcome to Supplement Tutorials. Today we'll be talking about, or we'll be continuing where we left from in our previous lecture. So today we'll be talking about the cellular phase of inflammation. And before we start, I would like us to hit on the subscribe button to subscribe to this channel where we give simple explanation to complex or sophisticated medical concepts. Alright, so before we start with the cellular phase, let's take a quick look at the initiation and the vascular phase of the inflammation we did in our previous lecture. Alright, so in our previous lecture, we defined what inflammation is. We said inflammation is the body's compensatory mechanism to get rid of pathogens or damaged tissues. And then we grouped the stages of inflammation into four. We said we have the initiation phase, the vascular phase, the cellular phase, and then the resolution phase. So in the initiation phase, we said that whenever an immune cell recognizes a pathogen or a damaged cells, it binds to it. And before an immune cell will recognize a pathogen, there's something we call immunosurveillance. And this immunosurveillance is the idea that immune cells are constantly patrolling, or they are constantly moving. The immune cells are constantly moving in the bloodstream. And this movement is mostly initiated or stimulated by what we call chemotaxis. So the chemotaxis allows the immune cells to propagate or to move within the blood vessels so that they can attract pathogens or damaged cells. Now, on the damaged cells or on the pathogens, there are substances or there are receptors that we call pathogen associated molecular patterns. And these pathogen associated molecular patterns could be anything that is part of the pathogen. It could be, for instance, in bacteria, it could be their flagellum, it could be their lipoproteins, it could be their teichoic acid, it could be anything a part of the bacteria. This same idea also applies to the damage associated molecular patterns. Okay, and on every white blood cell we have what we call pattern recognition receptor. So the pattern recognition receptor are found on the surface of of the white blood cells. And what this pattern recognition receptor does is that they bind to the pathogen associated molecular pattern or the damage associated molecular patterns. So and when this binding is done, so let's consider this to be, let's say, the flagella of a bacteria. So when this binding is done, when this PRR binds to this PAMPS, what happens is that it triggers a response, and this response leads within the white blood cells to release chemical mediators. And these are the chemical mediators we talked about. We talked about vasoactive amines, we talked about pro-inflammatory cytokines, we talked about lipid mediators, we talked about chemokines, and then reactive mediators. So in the vasoactive amines, we have histamines, bradykinins. In the pro-inflammatory cytokines, we have the tumor necrosis factor alpha, we have the interleukin 1, interleukin 6, and interleukin 12. And in the lipid mediators, we have the leukotrienes, we have the prostaglandins, and then the chemokines. Normally, the chemokines is usually applied in the cellular phase, so or during phagocytosis. Yeah, the chemokines are mostly what we call chemoattractants. So they mostly attract white blood cells to the site of infections. So we also have reactive mediators. So we have reactive oxidant species and we have nitric oxide. So these are reactive mediators. And then the lipid mediators, we have leukotrienes, we have the prostaglandins. So these chemical mediators triggers the remaining process of the inflammation, which is the vascular phase and the cellular phase. And this is what happens in the vascular phase, as we discussed in our previous lecture. So these mediators, especially the vasoactive amines and the reactive mediators and the lipid mediators, these are mostly the chemical mediators that affect the vascular phase. Sometimes we can talk about the interleukin 1, and then the interleukin 6 and interleukin 12 also having their influence on the vascular phase. But mostly it's the histamines, the bradykinins, the leukotrienes, the prostaglandins, and then sometimes the nitric oxide and the reactive oxidant species. So these mediators, what they do is that they interact with the endothelium. And once they interact with the endothelium, they cause series of events which include vasodilation. So we have the series of events here. We are not going to go through them details. The details is in our previous video. You can check that one out. So we have vasodilation, we have vasopermeability, we have leakage of complement proteins into the site of infection, we have swelling. And mostly the vascular activity or the vascular phase is what leads to the 5 cardinal signs of what? Of infection, of inflammation, as we discussed in the previous lecture. So we are going to talk about the cellular phase today. It's going to be a very wonderful lecture. Alright. So now we are in the cellular phase. The main aim of the cellular phase is to recruit leukocyte to the site of what? Of infection. And we have stages in the cellular phase. We have margination, we have rolling, we have adhesion, we have crawling, and then we have diapedesis. So let us take our time and go through how this process is initiated. Alright. So first of all, in the vascular phase, we realized that there was vasodilation. And we say vasodilation, there is an expansion of the what? Of the blood vessels. And this expansion of the blood vessels slows the blood flow within the vascular compartment or the blood vessels. It slows the blood flows. And once this slow movement is done, the WBCs or the white blood cells become sluggish in their movement. And they align themselves at the very edge of the endothelium. And this alignment is what we call what? Margination. So they are found at the margins of the endothelium. And this is what we call what? Margination. Now, in the vascular phase, we realize that the interaction of the pathogen recognition receptors and the pathogen associated molecular pattern triggers a series of what? Inflammatory cytokine release or inflammatory mediator release. And out of these mediators, we have tumor necrosis factor alpha, then we have interleukin 1. So take notice of this one. So this tumor's necrosis factor alpha and then interleukin 1 cause what we call endothelial activation. So we have margination. So the leukocytes align themselves at the margin. Then we have endothelium activation. So the endothelium activation, the tumor necrosis factor alpha and interleukin 1 interact with receptors on the endothelium, and this causes what we call endothelium activation. The endothelium activation leads to the generation or the change in the conformation of the endothelium, and this change in the endothelium conformation leads to the release of proteins or structures we call ICAM and then what? And then structures we call P-selectin and E-selectin. So P-selectin and E-selectin, so these are proteins that are popped up or that are released on the surface of the of the endothelium during endothelium activation. And what they do is that they interact with the ligands. These are what we call the ligands. These are ligands that are formed on the surface of the of the white blood cell. So these ligands binds to the P-selectin. So normally the P-selectin will come before the E-selectin. And what they do is that they seize the white blood cells and they cause it to roll. So the white blood cell moves from one selectin to another in a rolling manner. So it moves from one selectin to another and to another. So this process is what we call rolling. So we have our third process. We call it rolling. Our third process, we call it rolling. And then as the white blood cells begin to roll, these inflammatory mediators, interleukin 8, cause what we call activation of structures, cellular adhesion molecules we call ICAM and then VINCAM. So these are cellular adhesion molecules. And what they do is that they automatically immobilize the WBCs. They make them stop. So they immobilize the WBC, and that is why we call it the adhesion stage. So they immobilize them, and this is what happens. This interleukin 8 also cause the white blood cells to also generate proteins or glycoproteins we called selectins. So we also have selectins here. So these ones, so what they do is that they ensure what? Strong adhesion. So these ones, we have the ligands, then this one we have the selectins. So what they do is that they ensure strong adhesion to the ICAM and then the VCAM. So once this immobilization is done, it slowly cross the WBCs so that it will eventually what? Escape through the tight junction of the what? Of the endothelium. And this escape is called diapedesis. It's this form of amoeboid movement through the lateral side or the tight junction of the what? Of the endothelium. And these substances you see here are also adhesion molecules that interact with the what? With the WBCs during diapedesis. Okay, so we also have PE CAM here too. So this one too, we have PE CAMs here too. And these substances or these structures interact with the leukocytes as it undergoes the diapedesis. And ultimately, once this process keeps on going, it is recruiting more WBCs into the what? Into the interstitial space or the site where the inflammation is occurring, because the WBCs are needed at the site where the inflammation is occurring. So there should be more recruitment of these WBCs to cause or to further continue the resolution phase, either by undergoing phagocytosis, by engulfing pathogens, or by also clearing what? Dead or damaged tissue. So basically, this is what the cellular phase is about. It's a very brief lecture. In the next lecture, we'll talk about the phagocytosis, the process that involves in the phagocytosis. All right.